Well, hey AP, hope you're doing well when you watch this video. A little disclaimer, I haven't made a video in a little bit, so I'm probably going to be a little rusty, make many mistakes, but nonetheless, I'm making this to help you. I want you to know kind of what to expect as far as writing this cellular respiration lab report, filling out the packet. I also want you to have practice with data collection and data analysis. Of course, there's going to be tons of questions that relate to data on the test in May, so this is going to be good practice. So let's go ahead and jump in. So you have the cellular respiration, this table in your packet. And this table is based upon the first experiment. Now, we all did different experiments, of course, due to time, but germinating peas and dry peas plus beads and beads only. And that was, of course, the control with the beads only. So, instead of having you graph it on the packet, which is what they kind of talk to you about, I would like for you to have practice graphing in Google Sheets or Excel, whichever you prefer. It should work relatively the same. So you can see that some of our groups have not put in their data. The control group hasn't yet. Light has, because I helped them out. I had to finish the data collection for them, and I took a picture of it, so I needed to help them out. One group has. Good job, temperature. Well played. Looks great. And a few other groups haven't. Okay, so we definitely need control. We definitely need some other data points put in. So please get on that as soon as possible. And I've calculated the formulas in here, so hopefully they auto-populate the correct values, but you can double check them. So I wanted to walk you through, just with light, how to make a graph. Okay, so I'm probably going to make a few mistakes while doing this, and I'll probably have to correct myself, but nonetheless. So you kind of like highlight the data that you want to have, and click Insert. I'm sure you've done this before, but you click on Chart, and it kind of auto-populates a chart. That chart, of course, would not be the best chart to have for this. I mean, a bar graph, and what does these numbers even mean, and everything like that. So that is a problem. So let's not do that. Let's actually go over here and let's do a scatter plot. Okay, and so you can see the scatter plot automatically puts like time and oxygen consumption over here, and so you definitely need to have labels, and I'll go over that right now, how to figure that out. So if you want to do setup, customize chart and axis title. So chart title, make a good title. The title should kind of describe what's happening here. So a good title for this, an example would be effect of light on the rate of respiration. And some of those letters should be capitalized. And I think they did it in mung beans. I could be wrong. But you can see right there, good title. Summarizes kind of what the data says. Okay, and then you definitely need a horizontal axis title. You type this here, time, in minutes, and you do the same for the vertical title. Okay, so you want to have all those titles. Okay, you want to have the axis, the y-axis, the x-axis, and the title of the graph. Okay, so let's go back to setup here. And so you see we have like tons and tons of data points because we highlight a lot of data. And there could be an easier way to do this that you know of, but I just did it this way. We only need time and delta V, or the change in volume, okay? And so we don't need anything else. And so when I was kind of experimenting around with this, you just look at the column letters, and so I don't need C, and I don't need F, pipette reading, and I don't need that pipette reading. And so there you can see that we have our three, right? We have our three graphs, scatter point graphs for the different colors and time. Another thing that I want to mention right here is, like, you see how zero is kind of off the, I don't know why it does that. Again, you may know how to do this better than me, but I just went here to vertical axis and I did a -0.1, and then you can see how now we can see everything kind of happening. So that's helpful in the sense of that. So you can see series, that just is, we're going to click on that, and data series 1. And so you can make it different colors. I mean, for this one, why not make it purple, since that was purple light? And we're going to, for each series, I want to take my time with this, I want you to add a trend line. So the trend line is going to show you the slope, which you'll be asked to kind of find in other questions. What does the slope even mean? Let's label it. So let's use the equation here, and let's label it. So there's a slope, okay? And then use the equation. And then I also want you to show an r squared value. An r squared value basically allows you to evaluate the relationship between two variables. So in this case, time and oxygen production. R squared values closer to 1 indicate a more direct relationship. You can do a little research on R squared, but nonetheless, that's kind of what it means in general, okay? And so you can use the equation of the line to determine rate, since the slope of the line corresponds to the change in volume over time. So as you can see, there's a slope right there. Okay, and so then you want to change the graph line, go to yellow, okay? And then change, the different types of data collection and various things like that. So I want you to think about that and make all those trend lines. So again, we can make the trend line here. And let me see, there's a way to change the legend here. Let me see. The legend, and let's make it to the right. That's good. And then let's also have it labeling this situation. So how do I do that is the question. Let me see. Oh. So after pausing, I think I probably made a mistake and I probably should have highlighted opposite of the titles. So hey, first mistake, told you I was going to make that. So let's insert a chart again. And you can see, there we go. And so you can see how like the legend has words now. And so that's better. That looks better. Okay, so you want the legend to have labels. And so that's my fault. So let's go ahead and again kind of summarize through this again. Scatter plot, right? We don't want all the data, so you want to remove pipette readings. You want delta V, remove pipette readings here. You want delta V, etc., so on and so forth. Remember, of course, the first thing to make your chart titles, axis, horizontal and vertical axis. Different tiles for both of those axes or whatnot, how to polarize that. And so, so you want the legend to the right. So the legend goes, don't do an auto position. You want it to the right so it doesn't you can maybe drag it down, I guess, if you can. Yeah, nice and, okay, there you go. And so for each picture here, delta V, you can actually change the legend if you want. And I would suggest you doing this to not get confused. But right here you can see I can type in purple light and it should calculate on the graph, and it does. And so you can see that would be helpful to make sure your legend has a nice, good, description. And so delta V here, no light, and delta V here, green. Okay, and that way we kind of understand exactly what's happening in our data table, which is the point of this, right? Again, two data tables. And so let's go ahead and summarize one more time how to make a scatter plot. I'm just going to do it for one for time purposes, but you can see here setup, and if I'm going to click on this, kind of populates it for me, purple. So let's change it to purple for this experiment, for this graph. And you can see here, I want the trend line one more time, linear trend line of best fit kind of. Then you use the equation, and then I want you to show me R squared. Okay, and you do that for each, however many data points you have, you need to show that. Okay, again, my graph is not looking the best because the 0 is not really well seen. So you want to go down here to vertical axis and -0.1, and there, that's a better looking graph. Okay, and then you can have 3 different trend lines and all that. So for sure, reviewing one more time, I need a graph for the control. And again, this should be like the beans, the beads alone. And so if the beads change any volume, it's going to calculate that, but you have to minus, that's why it says corrected, because any change in the volume of the pipette for beads, that could have been like an atmospheric or some sort of change. So we need to address that. So that's what it means corrected. Just make sure that looks okay. So again, we have light ready. Here's temperature. We're missing a few other experiments. And so I need a graph of control and a graph of one of the other variables, okay? And so since we're all sharing this, I'm going to take this one away. And you can obviously collect this data. Don't wait for someone to do it, because they might not have done it correctly. Make sure you check it. Make sure you do it on your own. whether you want to create a new graph or copy it. I would just cut and paste it into a document and print it out when you're done, and that way people don't steal your work. Okay, so I'm just going to delete this, and you see you can like copy the chart, right, and then like paste it into various things, but just hit delete on that, and there we go. Okay, so going back to the packet, do that instead of graphing it by hand, and then from your graph calculate the rate of oxygen consumption. So this is just kind of the slope, or if you're wondering how to figure it out, it's delta y over delta x. You can plug in the data there, figure out the slope for the germinating seeds and the dormant seeds, okay, and then see what the difference is with that. And then it asks you to make a claim. Of course, the germinating peas, we expect to have a higher rate of respiration. And why is that? Why would that be? What's happening to the pea? Why are their energy needs? What's happening as far as, macromolecules or starch or cell division, mitosis? You need to talk about some of that in that answer, okay? Then it goes into the ideal gas law. And you can read it right there. I'm not going to read it for you, of course, but the next question is, using the ideal gas law in your experience in this lab, give the variables that had to be controlled for your data to be valid. Okay? Well, we had to control temperature, and we also had to control pressure. Okay? The water baths were used to control, of course, temperature, and then pressure was controlled by leaving the respirometer tips open to any atmospheric pressure. So any changes in the pressure was registered by the beads only. Okay? And so look over that law. Again, you don't have to memorize any equations for the test in May, but look over that and then answer those next two questions. It's pretty straightforward. Then for some of you, you did a experiment. Again, we're all sharing data here to learn the most that we possibly can. And so your lab goes into question hypothesis. You can fill that in. Hypothesis, write that. Independent, dependent variables, control group, that's great. And then you don't have to worry about equipment. Let me see if I can write here. You don't have to worry about, nope. I don't know how to, nope, that's not how you write. You don't have to worry about equipment or anything, so cross that out. You don't have to worry about procedure. That's kind of beyond the scope of this assignment. Safety steps you don't have to do, collecting data, all the rest of that you don't have to do, okay? So what changes or modifications have you made? Did you have to adjust anything? Wrote results, did you see? Summarize what you saw if you did. In either experiment, summarize what you saw. Even if you just did the control experiment, summarize what you saw. Accept or reject your hypothesis. Again, we wrote an alternative hypothesis, not a null hypothesis in this. And then what sources of error? Of course, always error, human error, various things like that. Skip the rubric. And so you can go down here to this big idea assessment, okay? So these questions are going to help me understand if you truly kind of gathered some knowledge as far as what's going on in this lab. So the first question deals with the group does not add any potassium hydroxide. We discussed in class why potassium hydroxide was added to this experiment. So why would you expect their group falsify their data? I mean, their data looks pretty much like our control. Why would that be a problem if they didn't add any potassium hydroxide, okay? For number 2, it's asking you to investigate heat, okay? A simple apparatus and experiment that will allow you to measure the respiration rate of germinated and non-germinated peas using temperature of the peas themselves, okay? So it's not exactly the same as what one of the groups did as far as temperature of the water. So in order to measure temperature as a function of respiration, the pressure, volume, and kilomoles must stay the same. So we shouldn't add potassium hydroxide in this because the volume of gas should stay constant because we're investigating temperature. And if you look up here, temperature is one of the variables. Oh man, scrolling. There we go. So you can see temperature of the gas right there. And so pressure of the gas, volume of the gas. So we got to keep all that the same if we're investigating temperature. So you want to not add potassium hydroxide. And I'm not going to tell you why because that's the answer to number 1. And so number 3, Of course, just some math there. 7.3 calories per mole of the hydrolysis of ATP to ADP. Here's the complete oxidation at 686 under standard conditions. So just calculate the efficiency of total ATP production from 1 mole of glucose in the organism, okay? So how many ATP do we make really quick? Well, roughly 36. 36 right there is roughly what we make. So that should help you understand the math there. And then they give you this almost kind of like a free response question, which is really awesome. And so I really don't want to help too much with this. Basically, they're asking you to figure out, read the legends, read the keys. Obviously, note the standard error bars if they overlap. Something that means something. If they don't overlap, that means something else. We talked about that in class. And yeah, it's a really good. Obviously, it's directly from a peer-reviewed article. So go through and answer these questions, and then just trying to show big picture, big ideas, get your thoughts down on paper. And then number 5, common ratio used in storage is the respiratory quotient. That's fun to say, respiratory quotient. And so with that being said, you can see that they give you like this little equation here. And so explain why the respiratory quotient for the oxidation of carbohydrates should equal 1. And so the complete oxidation of glucose requires 6 moles of oxygen and yields 6 moles of CO2. So if you kind of think like volume of CO2 released, volume of oxygen produced, 6 divided by 6, that's just the answer to letter A to kind of help you with letter B. Okay? And then finally, another practice for data. Researcher wants to examine the effect of temperature on bedbug oxygen consumption. To do so, she collects 100 adult male bedbugs of identical mass, that's impressive, from her laboratory bedbug colony and incubates 20 in each of 5 different temperatures. Okay, so independent, dependent variables, of course. Mean oxygen consumption plus standard error for all treatments is described in the table below. So you can see there, that, again, the standard error bars. Might want to refresh that, what that means. We talked about it in class. They provide more information about how different the means may be from each other. And so you definitely want to graph. When you graph this, they're going to ask you to graph it. Graph the standard error bars with like the lines, right? Like graph the line and then put the vertical line up and down, plus or minus however much it is. And then answer a few questions, and that is your cellular respiration lab. Okay, that's another page. So let's review one time. I want your 2 graphs, control and whichever other experiment that you did, or if you did the control, pick another experiment to do. I want these questions answered in complete sentences. Again, you don't have to do all the methods and procedures and everything like that. Definitely looking at the assessment. This is going to be worth at least 3 or 4 in the lab category. If you have any questions, please let me know. Hopefully this was helpful. Hopefully my mistakes didn't mess you up too bad. But I'm going to try to pause the recording here. And yeah, let me know if you have any questions. Thank you so much for your time.